Hey guys, welcome back to One Stop Biology. So guys, in today's video we are going to cover translation. So I hope you remember what all topics we have covered in this chapter. We are right now at molecular basis of inheritance, which is the second chapter of unit of genetics for class 11th NCERT. So guys, I'll again repeat that we are covering all these chapters as per the NCERT syllabus because right now we are running the NCERT series. Now before I start off with translation guys, I would like to remind you to definitely like the video and share it with your friends if you do understand the topic completely once you complete this video. And also your feedback is very important to me, so please do not forget to let me know in the comment section whether you like the video or not, and if you have some doubts, please do let me know regarding your feedback so that I can improvise on the videos and make it better for you. So let's start with translation. Now guys, translation refers to the process of polymerization of amino acids. And what does these amino acids form? They form a polypeptide chain, right? So basically I'll again draw the central dogma that we studied in the very first video, which is DNA. It keeps on replicating, then it forms RNA, and this RNA forms proteins, and three processes are involved. First is replication. So DNA replicates itself entirely. Then when it changes to RNA, it is transcription. And then when this RNA forms protein, it is translation. So this is the central dogma, right? Now this process is basically translation, where RNA changes into, RNA codes for, RNA gives the genetic information in the form of proteins. So basically there is polymerization of amino acids to form a polypeptide chain. Now how is the order and sequence of amino acid defined, or how is it decided? So basically it depends on the sequence of bases in the messenger RNA, the RNA which is going to be translated. So as per the sequence of nitrogenous bases in the messenger RNA, the sequence of amino acids will be defined as per that, right? Now these amino acids, the amino acids are joined together by peptide bond. They are joined together by peptide bond. So say two amino acids are joined by peptide bond. Now these, formation of these peptide bond basically requires energy, right? So that is why in the very first phase, amino acids are activated in the presence of ATP. Remember, the amino acids are activated in the presence of ATP, and these are linked to their cognate transfer RNAs. Now I hope you remember the structure of transfer RNA that we studied in the last video as an adapter molecule, right? Transfer RNA works as an adapter molecule, and it joins to a particular amino acid, and there are different transfer RNAs for different amino acids, right? So that is why these actively charged, basically these activated or charged amino acids are linked to their cognate transfer RNAs, and this process is known as charging of tRNA, transfer RNA, or it is also known as amino acylation of transfer RNA. It is also known as amino acylation of transfer RNA. So now if two such charged transfer RNAs are brought close enough, the amino acids present on these tRNAs form a peptide bond, right? So they form a peptide bond. And to enhance this rate of peptide bond formation, one would require catalyst as well. right? To enhance this performance, one would require a catalyst as well. Now guys, basically, what is the cellular factory which is responsible for synthesizing proteins? Now the cellular factory responsible for synthesizing the protein is, you know what? It is ribosome. It is ribosome. Remember that it is ribosome. Now these ribosome consists of structural RNAs. These ribosomes basically have structural RNA and they have about 80 different proteins, right? Now these different proteins are in its inactive state. These 80 different proteins are in inactive state, right? And these proteins exist as two different units. So you basically have a larger unit and a smaller unit, right? So you have a larger subunit and a smaller subunit. When this small subunit encounters a messenger RNA, it encounters mRNA, the synthesis of proteins or the translation of protein begins. And there are two sites in the larger subunit. In this subunit, there are two sites, remember, for subsequent amino acid to bind to, and thus it is close enough to have peptide bond as well, right? So basically the ribosome, this entire is nothing but the ribosome, right? This is the ribosome. Now the ribosome acts as a catalyst as well, right? And catalyst for what? For formation of peptide bond, right? Now a translation unit in mRNA is nothing but sequence of nitrogenous bases. Now these sequence of nitrogenous bases have three things. They have a start codon, which is nothing but AUG, which codes for methionine as well. They have stop codon to stop translation, and then they have codes in between for polypeptide, right? So basically they have a chain where they have a start codon to start translation, they have a code for polypeptide, they have a code for translation, and then they have a stop codon to stop translation, right? Now apart from that, the mRNA also has some additional sequences, and these sequences are not translated, and these sequences are known as untranslated regions. In short, they are known as UTR. So the mRNA also has untranslated regions which are not translated, and these are present at both the end. They are present at 5 end, which is the start codon wala end, and they are present as the 3 end, which is the stop codon wala end, both the ends. And they basically, make the translation process more efficient. They make the translation process more efficient. That is why they are present. It's not that they have no reason to be there, right? Now let's see what happens during translation through the diagram that I showed. So let's understand translation through this diagram. So I'll show you again, guys. Basically, this is the ribosome, right? So this is one unit, the larger subunit. This is the smaller subunit of ribosome. This is the smaller subunit. This is the larger subunit, right? Now for initiating translation, what happens is the smaller subunit of ribosome binds to the mRNA. So this is nothing but the mRNA strand, right? So this is the mRNA strand. Now the smaller subunit of ribosome binds to the mRNA, right? And where does it bind? At the start codon, AUG. Now this AUG is recognized by initiator tRNA. This AUG, right, is recognized by initiator transfer RNA. So when we studied transfer RNA, we studied that there is an initiator tRNA, but there are no terminating tRNAs, right? There are no tRNAs for stop codons. There are, however, they are our initiator transfer RNAs, right? So these are recognized, the start codon wala sites are recognized by initiator tRNA. Then the ribosome basically proceeds to the elongation phase of protein synthesis. Now during elongation phase, during this stage, what happens is complexes are basically composed of amino acids which are linked to tRNA, right? They keep on binding to the appropriate codon in mRNA and form complementary bases with the tRNA anticodon. So, right? So basically, if you see here, this region, right? This tRNA is coming and binding to this region, right? So it complements the base with, say, A, G, G. This is nothing but complementing this. In the same way, we have this, right? So complementing this, you have U, U and A. Now this binds to the appropriate position, translates, and what you are seeing here is a growing polypeptide chain, right? So these, what they're doing is basically these transfer RNAs are coming, binding here, they're translating. So basically they're specific to a particular amino acid, they're translating, right? Now what is happening is the ribosome moves from codon to codon along the sequence which is basically dictated by DNA, right? This mRNA sequence is not going to change. The ribosome is just helping in translating the sequence, right? So this keeps on translating sequences in the same way. And at the end, what happens when the stop codon comes, right? So there are three kinds of stop codons, right? So when either of the three of the stop codon comes, a release factor binds to the stop codon, which basically terminates translation and releases this polypeptide chain. So this polypeptide chain completes. So here one transfer RNA is coming, translating into valine, and this is basically joining by the polypeptide chain. Again, it will go, another transfer RNA comes, it will translate it another amino acid, it will basically join by peptide bond. Now once you have the stop codon, once the stop codon comes, once the translation is complete basically, right? The translation of that genetic information is complete. You have this one of the stop codons, and as soon as the stop codon comes, there is a release factor which comes and combines to the stop codon. This release factors comes and binds to the stop codon. And as soon as this release factor binds to the stop codon, the translation is terminated. And this completed polypeptide chain is released from the ribosome, right? So this is the entire process of translation, guys. And again, you see we have new factors involved. We have a very important factor involved here, which is ribosome. Now, without ribosome, translation is not possible, because once the mRNA comes to the ribosome, translation starts, right? So this is the entire process of translation, which is nothing but polymerization of amino acids, which is making a polypeptide chain, right? Translating into a polypeptide chain. So guys, I hope you have understood the process very well. I'll tell you again, these topics are very, very important from boards and NEET point of view, right? So I hope you have understood it very well. I would request you to go back and read this again from NCERT and come back and comment on this video if you have any doubt. But please do let me know if you have any doubt. You can either message me in the comments of the video, or you can also message me over WhatsApp. I have given the WhatsApp number in description of the video, but please do message me if you have any doubts at all. And yes, if you understood the video very well, please do not forget to like it, right? I at least deserve your likes. So please do not forget to like it, share it with your friends. And yes, if you are new to the channel, please do not forget to subscribe to the channel to get all the updates, right? So guys, let's make sure that we have completely understood this topic and revised it again and again. So see you in the next video, guys. Bye bye.